So my name is David Elliott. I'm a microbiologist. I've done my PhD in microbiology at University College London. Now I'm working here at MMU on environmental microbiology, and one of my latest projects has taken us to Kalahari, which is a desert or an arid area. Arid areas cover about 1/3 of the Earth's land surface. Plants don't do so well in these places because they need water. And so we have very big interspaces in between the plants. And in these interspaces, what most people would just tread on think is sand, doing nothing. The sand is colonised by microbes. They colonise it to such an extent that they make the sand go hard and become stabilised. And we call this a biological soil crust. And my research in the last few years has been looking at the microbes that live in these biological soil crusts and trying to figure out some of what they do and trying to figure out what is their impact or their role in regulating the carbon cycle. The biological soil crust is actually very strong. If you pick it up, it can remain intact, even quite a large area of crust. And it's held together by these strands of cyanobacteria. I broke that up, quite laborious actually, with scalpel. Again, using careful technique not to contaminate with my own microbes or the microbes from the other parts of the environment. And in the end, we get about 1/4 gram of sand into the DNA extraction kit. And then at this stage, then we start the process, which involves really shaking and physically disrupting the sample. And the cells end up smashed up into a tube like this, and the DNA goes up into the liquid. But also we've got lots of other bits of the cells, like the cell wall is up there, and any bits of muck that was in the soil is all there. So we put that in the centrifuge, and that pushes everything down to the bottom using about 10,000 G of equivalent of gravity. And in the end, we end up with some clean DNA that looks like that, just looks like water. There's a few methods that we can use, but the method of choice now is to use a high throughput DNA sequencing technique. That relies on a technique called the polymerase chain reaction, or PCR. And what PCR does is it will amplify a short stretch of DNA which you know something about. So in my case, it's bacteria and fungi that I was looking at. And they have special genes called ribosomal genes. So all of fungi have areas next to the ribosomal gene which always have the same sequence. So we can use this as a hook, and we amplify the bit of DNA of these ribosomal genes using the polymerase chain reaction. The possibility of doing high throughput sequencing really came on the stage around 2008 or 2009. Before that, the maximum you could hope for in a well-funded study would be maybe 2,000-3,000 sequences for the whole study, really. Now we can get 3,000 sequences per sample, which means hundreds of thousands for the whole study. And it's really revolutionised our ability to measure, quantify and describe microbial communities. I've had a really long-standing interest in computers, and for me it was very natural and really very welcome actually to have this massive data set and have the opportunity to really explore it and learn how to do the bioinformatic techniques. So I've really enjoyed doing that. I'd like to make microbes visible, make them sort of, because as it stands, we look at soil and it's just brown. The plant relies on the soil, but not only the soil, it relies on the microbes in the soil. And that's not normally visible. And these new sequencing technologies that we're using is allowing us for the first time to really have a look into the microbial diversity of the soil. So first of all, I'd like that to be sort of recognised not just by scientists and not just by the public, but by policymakers who are making decisions about how to manage land. I think they should know about the microbes that are there, even if we don't yet have the full knowledge of the function. We should know that they're doing stuff and that it's worth finding out more about what they're doing, especially in relation to their symbiotic relationships with plants.